Hello and welcome to today's Byte-Size Genome Informatics webinar regarding the General Feature Format 3 or GFF3 files. These are files that I commonly use to describe features in genomes such as gene models, gene annotations and so on. Before I get into the content of the webinar, I'd just like to go over a few things regarding who I am, the types of activities I'm involved with, and how you might look to engage me into the future. So my name's Nathan Watson-Haig. I'm a bioinformatician of around 20 years. My main fields of expertise are in genome informatics, high performance computing, Linux systems administration. I do pipeline development and optimization. I have a wealth of experience with SnakeMake, particularly looking to analyze large wheat and barley data sets, amongst others. I have quite a number of years of experience in bioinformatics training, and my programming languages of choice are Bash, Perl, R, and so on. I also have some experience in other languages such as Fortran and JavaScript. However, usually I require a command reference of some sort to sort of make some headway there, and maybe not as quick as other people. The types of projects that I've been involved with in the past are around genome assembly and gene annotation, hence this webinar. I've also been involved in a lot of variant analysis, and I had a PhD in phylogenetics and molecular sequence analysis. I've been involved with transcriptome assembly and transcriptomics and differential gene expression analysis. I've also been involved with projects involving CRISPR editing and characterization of CRISPR edits in wheat and mouse ESLs. I've also been involved with looking at genome diversity, and I have a keen interest in variant graphs and genome graphs and things as representations of genetic diversity amongst populations. And I have recently founded a business called Genome Informatics and Bioinformatics Training. You can contact me at this email address here, or you can have a look at the LinkedIn company page down here. The types of work that I'm involved with is freelancing work, so predominantly around sort of pipeline development or analysis of data sets. I also provide consulting advice around sort of experimental design, the types of analysis that might be appropriate for a project and interpretations of things. I'm also involved with developing and delivering training. That might be one-on-one training, or it might be training with small groups of people or labs. And these sorts of training events can take place either online, as is the case with most things at the moment with COVID-19. However, as things tend to improve, this training will move more towards in-person training. Now, the concept of this bite-sized genome informatics webinars is that they will be held regularly. So at the moment, it's weekly, this time every week. That's Thursday, 1 p.m. Adelaide time, which is just nicely situated so that people overseas from India through to Japan, sort of time zones can attend, and it's a reasonable time for them. They're short, so 30 minutes, and they're focused. That is, they're centered around a well-defined subject matter, such as this one, which is the GFF3 file format, and they're generally free for non-industry, or there's a small charge for industry attendance. And you can have a look at the events that I have available at this ticket tailor address down here. So the key learning outcomes for today's webinar is that hopefully you'll go away at the end understanding a little bit about what a GFF3 file is and what it's used for, the basic structure of a GFF file, and so you can start to manipulate and modify it and do various other operations on it. I'll show you how to validate a GFF3 file and why that's important. I'll also show how you go about compressing a GFF file to allow fast coordinate range queries, which are really important, especially when you want to start extracting out features from different places in chromosomes, or whether you want to visualize the GFF file in a genome browser or something like that. So a genome is packaged up in a cell nucleus. I'm going to show you this schematic cartoon of a imaginary chromosome. This could be from any species, I guess. In this case, I've labeled it chromosome 1. And on this chromosome, we have a gene model. And in this case, I've labeled this mRNA 1. Now, when we think about annotating genomes, we are really, what we're doing is we're annotating features, that is chunks, coordinate ranges on the chromosome. That is something that lies on a given chromosome with a start position and an end position. So it's a range. And when we look at these messenger RNAs or gene models that are annotated in genomes, we can decompose those down into individual features. So here I'm showing you the exons. There's 4 exons in this particular messenger RNA. The first exon here coincides with this UTR, the thin bit here. And an exon, by definition, is the coding sequence as well as the UTRs. Some people think that it's just the coding sequence, but it's not. And so you can see here we've got 4 features or 4 exons, and each one represents a continuous range with a start and end position. And the same can be said for different aspects or different features of this messenger RNA 1. So we have introns, which can also be described as a range with a start and an end position. The same for the coding sequence, and the same for the UTRs. So what I'm trying to do here is just to get you thinking about how these gene models can be broken down into just individual features, that is exons, introns, coding sequences, and UTRs. This is important for how the GFF3 file works and how you structure it, because it contains this information, and it's a feature-based file format. Each row in a file contains a feature which describes its location, and as well as some other aspects and properties. We'll get onto that in a moment. So here we can provide some coordinate space for the chromosome. So in this case, the chromosome goes from one base up to 2 kb. And we also have some markers in here for 500, 1 kb, 1.5 kb, just so you can see where these exons are roughly. And if we start to label up these exons in terms of their start and end coordinates, the first exon here starts at 300 and ends at 550, and then we have the other remaining 3 exons with their coordinates start and end. One thing to bear in mind at this point is when we're talking about the start and end coordinates, are these positions inclusive or exclusive of the position of the base at that given position? The reason this is important is because there's multiple coordinate systems that can be used for describing coordinates in a genome. So let's take a closer look at that. So with this particular example, we're just looking at a mock sequence here, 7 bases long, and in the middle here is a start codon ATG. And we want to describe the location of this ATG codon within this 7 base sequence here. So as I said, there's 2 systems for this. One of them essentially starts numbering bases from 1 and numbers the remaining bases sequentially up to 7. And this system is called a 1-based system because it starts at 1. Now the feature in the middle, this ATG that we're looking to describe as coordinates, can be described as having a start position as 3 and an end position or coordinate as 5. And these 2 positions are inclusive of the start and end of the position. And this has a consequence when it's when we're calculating the length, because the length is equal to the end minus the start. And in this case, we have to add 1 because 5 minus 3 is 2, and we need to plus 1 to get the length of this feature, this start codon of 3. Now, this type of coordinate system is called the one-base fully closed system. Fully closed being that the coordinates used to describe the feature, the start and the end, are included within the feature itself. Now, there's another system that's used, which starts numbering the bases at 0 and numbers the rest sequentially up to 6. And this is called a zero-based coordinate system. And in this system here, the start and the end coordinates are slightly different. So the start here is the same, but it's because it's zero-based, the start is number 2. So again, this is including the first base of the feature. However, the end coordinate is actually specified as the position beyond the feature that we're looking at. So in this case, we say that the end coordinate is 5, and this end coordinate is not inclusive of the base at that position. So this coordinate system is called a zero-based half-open coordinate system. Now, the reason for having some of these things might become a little bit more evident as you look at how you calculate the length of such a feature in this coordinate space. And you can simply calculate the length as the end minus the start. So 5 take away 2 is 3. So it's a 3 base pair long feature. And these two systems are used by these two different file formats. So the one-base fully closed system is used by the GFF3, the subject of this webinar, whereas the bed file format, which will be the subject of a future webinar, uses this zero-base half-open coordinate system. This is really important when you're looking at files and even when you're sort of doing transformations between GFF file format and bed format and trying to reconcile those, because you can very easily get off by 1 errors in the conversions. So now we've had a look at those coordinate systems. We're using this one-base fully closed system here for the GFF file. So here I've numbered the exons 1, 2, 3, 4. So here this gene by inference is on the positive strand, the forward strand. And we're going to take a look at how these exons can be described as a GFF in GFF3 format. So a GFF3 file is just simply a plain text, tab-delimited plain text file with 9 columns with these different types. So the first exon here, exon 1, with a start of 300 ends at 550, we can describe like this. So sequence ID is the chromosome on which that feature lies, and we've got the start and the end positions here. We've also got the strand here, the forward strand, the positive strand here, and we've got an attribute here which is simply a tag equals value pair. You can have multiple attributes and they get separated by a semicolon. So in this case, ID, this has to be unique within the scope of the GFF3 file. And then we have a few other columns. So source here, that is the source of this feature. Who came up with or defined this feature. And so in this case, it was me, so I've just put my name in. Source is a free-form text field, so you can essentially put anything you want in there. However, type is a tightly controlled field. This type has to, for a valid GFF3 file, has to be a feature that's described in the sequence ontology and a sequence ontology. It can either be the name of the term, or it can be the actual accession and number. We'll come on to that later. Score is not relevant for this type of feature, so it just gets a dot, and same for phase, just a dot. And we can do this for the remaining exons, and you'll notice the identifier of the exons in the attributes column is unique. Now at this point, there's nothing that links these exons together. They're just 4 different ranges in chromosome 1. There's nothing that links them together in any way at this point. So we need to include an additional feature in here that then links them together and infers a parent-child relationship. So we include a new feature in here, an mRNA feature type. It has a start and end coordinates, which is basically fully encloses these 4 exons. And it, again, has its own identifier. And the exons themselves then start to get some additional attributes separated by this semicolon. In this case, they have this parent tag value pair. And this is where the parent-child relationship gets expressed. So each of these 4 exons is a child of this mRNA feature. Now there are other types of features that we might want to include in here to make it a bit more complete. So here I've included a gene feature, and the messenger RNA is a child of that gene. Now, the reason why you'd want to have this type of relationship here is that you might have a gene that has multiple splice forms, multiple messenger RNAs, and you can include multiple mRNAs with this sort of relationship by including the gene feature type. And to make this more complete for this particular mRNA up here, I've included a feature for the 5' and 3' UTR, as well as the coding sequences that are present in each of the 4 exons. To make this a valid GFF3 file, it must contain this GFF3 Pragma. Pragmas are these special strings lines that start with a double hash symbol, and it's this particular one has to be the first line. It denotes that the contents of that file is a GFF3 file, and can be used for validation of the file that it meets the requirements of the GFF3 version. Now, in here, all we know is that these features are on this sequence called chromosome 1, and they have coordinates, but we don't know how big chromosome 1 is in this GFF3 file. So what happens if we had a feature where the start or the end went beyond the limits of the chromosome, either before the start of the chromosome, or in this case, that sort of went beyond the end of chromosome 1? If we had features like that, we might be interested in actually capturing whether we have features that lie beyond the chromosome, because those are likely errors that we want to catch. And when it comes to validating things later on, there is a pragma that we can include in here to help with doing that validation, and it's called the sequence region pragma. So the sequence region pragma has the chromosome identifier, and it has the chromosome start and end position. And when you include this information in your GFF3 file, and when you do run a validation of the file, the validation will check to ensure that all of the features defined within the GFF3 file fall within the limits of this chromosome. And if there's any features like this one that lie beyond the end or the start of the chromosome, it will flag that as an error. So I talked about validation. How do you actually go about doing that? Well, one of the first things to think about or look at is this sequence ontology browser. This is the sequence ontology specification down here that has lots of information about what's a valid GFF3 file and how things should be structured, what the different columns can and can't contain, etc. The reason why you actually want to do some validation is that tools that interpret these features in this GFF3 file expect it to conform to certain rules or to conform to the specification as set out in this link at the bottom. If it doesn't conform to those rules, what might happen is that you try and visualize that GFF3 file in a genome browser and it might not render correctly. Things might get really screwed up, or when you start using command line tools to extract things out, you start having to fudge certain things because they're not as they were intended, or someone's used and abused the specification beyond what it should have been used for, and so it can cause all sorts of problems. It's really important in my mind to validate GFF3 files, and it's really surprising that many GFF3 files distributed as part of large genome sequencing consortia don't actually validate. Very, very, very often, um, most GFF3 files actually miss this GFF version pragma at the top. Quite often that may just be the only thing that's missing, and you can just include that and it validates fine. The other thing that is a very commonplace is this attribute capitalization. So one thing that I didn't mention, but is mentioned in the specification, is that all attributes in the column 9 are case sensitive. So that means ID uppercase is different from ID lowercase and is different from capital I little d. And the other thing to note is that all attributes that start with a capital letter have been reserved by the specification. And so if you want to include your own tag value pairs for something, you can't use a capital first letter for that tag name because that's reserved by the specification. You have to use lowercase. And there's many times where these files are distributed that use an uppercase first letter. Quite often, CDS features have an incorrect phase, and I'll describe that in the next few slides. The third column, this type column, is quite often that they have incorrect types. That is, they are not strings from the sequence ontology or not sequence ontology accessions. So for instance, the sequence ontology term CDS uppercase is correct. And so quite often you'll see things like lowercase CDS, which is not correct. And you'll also see various features. So we've just had a look at, when we define a parent attribute for a feature to define a parent-child relationship. That relationship can only happen if those two features types have a parent-child relationship in the sequence ontology. And so if you try to enforce a relationship between two feature types that are not related in that way in the ontology, then that breaks the validation. So as I mentioned, phase in this coding sequences is a common issue. What I mean by this is if we have a look at, say, two coding sequence features, so the green boxes here, coding sequences result in proteins, and we have open reading frames in coding sequences once you've got the intron spliced out. However, the phase of a particular coding sequence depends on essentially the multiples of 3 that's within the coding sequence. And in this first coding sequence here, the first full codon here, it starts at the very first base. So this gets a phase of 0. And the second coding sequence feature here, once you've spliced out the intron, will have its first full codon here, starting at the first base as well in that coding sequence. So that gets a phase of 0. However, that doesn't have to be the case, because if you have coding sequence that look features that look like this, and in this first codon sequence, you have the first codon starts at base 1 in that coding sequence, and so it gets a phase 0. And in the second coding sequence feature, the actual first full codon starts at this third base, because once you splice out this intron here, we have another codon CCA in the middle of the coding sequence. And so the first full codon within this CDS feature starts at this third base. And so this coding sequence feature gets a phase of 2. And for completeness, we have another situation whereby the first full codon in the second coding sequence feature starts at the second base of the coding sequence, and this gets a phase of 1. So if we run this tool through this Genome Tools Validator, so this is a website where you can upload a GFF3 file to do some validation. If you upload this particular GFF3, it will complain about the phase of these coding sequences here, which were just dots, and it will suggest that the phase for each of these should be 0, 0, 2, and 1, respectively. And so these tools this genome tools can suggest and tidy up simple errors in your GFF3 file. The other thing that's checked when you do validation of your GFF3 file is this type column, this third column here, which I said has to be one of the terms in the sequence ontology. You can view the browser here and navigate through that and search for various terms. So that has to match the terms, and it's case sensitive, as I said before. And it could also be the sequence ontology accession. So in the case of the CDS features, it could be sequence ontology : 0000316. That's not so human readable, and so most people will just use the term, the sequence ontology terms such as CDS, so it makes it more human readable. So how do you go about doing validation? So here's an Askinima recording of doing some validation of some common GFF3 files that are available from standard genome sequencing consortia and genome consortia. So this one is from the Arabidopsis thaliana TER10 gene annotation file. So we're just downloading this file now, and we'll run the GFF3 validator and doing a type check on that third column for the sequence ontology. And the first thing it comes back with is that it doesn't have this GFF3 Ragma here, so it doesn't validate. Now we're going to have a look at this Bali version 2 gene annotation file for chromosome 1H, just to see whether that validates. So these are all files directly from those consortia. And again, we'll also include a type check on that third column. And here we've got an error in the validation that this type feature with this type pseudogenic transcript has a parent-child relationship with another feature called gene, and that's not something that can be inferred by the sequence ontology. It's an incorrect relationship there, so that fails validation. We'll have a look now at the wheat, the IWGSC version 1.0 high confidence genes, and have a look to see whether they validate. Again, doing a type check on the sequence ontology column. And again, here we've got another example where there's no GFF version Ragma. So just for completeness, let's insert this with sed on the command line and insert the Ragma at the start and see if we can get GFF3 to validate. What we get at the end here is that it does indeed validate. There's correct GFF3, but there's a bunch of warnings that basically say that there is this sequence region Ragma missing. If you remember from what I said earlier, this is where you define the start and end position of a chromosome, and that can help in validating that features fall within the correct coordinates or the correct size of a particular chromosome. Okay, so moving on to compression of GFF3 files. This is important, as I said, if you want to pull out features quickly from a file, especially if the GFF file starts to get quite big. Performance, especially genome browsers and things like that, degrades very rapidly with increased size. And if you don't have it indexed, the browser generally has to load the entire file into memory in order to do things reasonably quickly. However, it's best not to go down that route and best to know how to compress and index files correctly. So the solution that we need to take here is to do compression using a couple of tools from the htslib package, one executable called bgzip and another called tabix for doing the indexing. However, prior to the indexing, we probably also need to do a GFF3 sort to make sure that all of the features are sorted correctly for the indexing to succeed. So here I've got another Askinima video to show you the compression and indexing of some of these files that we've already looked at. So again, here we'll look at the Arabidopsis TER10 gene annotation file. So we'll BGZip compress it with 4 cores here and output it to a new file. And then we'll try and index it. And it says, it complains it fails unsorted positions. So let's try and sort it with GFF3 sort. We'll do this on a pipeline. So we sort it, pipe it into BGZip4 with 4 cores for compression. And so this file here now should be sorted and compressed. And we'll then index it, and it shows no error. So we successfully indexed the file. Now we'll have a look to see whether that file validates. Again, doing a type check for the sequence ontology column. And we see here that there's an error, again, saying that there's no GFF version pragma. So just because we can index a file doesn't mean that it's valid.